Yeast and bacteria are both used to make fermented food, but they're not just slightly different microorganisms. They're about as far apart biologically as you are from a mushroom. And that difference, at the molecular level, is the entire reason why beer tastes like beer and sauerkraut tastes like sauerkraut. In this video, we're going to go through exactly what sets them apart, and why understanding that changes how you think about every fermented food you've ever eaten. Let's start with the basics. Bacteria are prokaryotes, some of the simplest life forms on Earth. They have no nucleus, no complex internal structure, just a cell membrane, some DNA floating around inside, and a handful of proteins doing all the work. Everything happens in one single compartment. Yeast are fundamentally different. They're eukaryotes, single-celled fungi, yes, but in the same domain of life as plants, animals, and humans. And what that means practically is that a yeast cell has compartments, specialized structures each doing a specific job. There's a nucleus that stores and protects the DNA, mitochondria that act like tiny power plants converting nutrients into usable energy, and an endoplasmic reticulum that functions like an internal transport and manufacturing system. You don't need to memorize these names. The point is just this. A yeast cell is genuinely more similar to one of your liver cells than it is to a lactobacillus bacterium. They look completely different under the microscope, but structurally they're playing in the same league. Even how they reproduce is different. Bacteria clone themselves through cell division. One bacterial cell splits into two identical copies. Yeast do something called budding. They grow a small daughter cell that eventually detaches. There's one exception worth mentioning, Schizosaccharomyces, the only yeast that actually divides like bacteria. We'll come back to that one another time. Now here's the thing that unites them despite all these differences. Both bacteria and yeast have one fundamental goal: survive, multiply, and pass on their genetic code. And to do that, they need energy. That energy comes from sugar. Both organisms convert sugar into ATP, the universal energy currency of life. Think of ATP as a rechargeable battery that powers everything happening inside the cell. But here's where it gets interesting. To produce ATP from sugar, the cell runs the sugar through a long chain of chemical reactions, what we call metabolism. And this chain needs to be continuously recycled. At the end of this chain, there's always a final product, something the cell essentially has to dump because it can't use it anymore. And this is where yeast and bacteria fundamentally diverge. Classic brewing and wine yeasts, the Saccharomyces cerevisiae strains that humans have used for thousands of years, primarily produce ethanol, glycerol, or acetic acid as their main end products, depending on the conditions. That ethanol is why your beer has alcohol. The glycerol contributes body and mouthfeel to wine. Later in this series, we'll look at non-Saccharomyces yeasts that run completely different pathways and produce a much more complex spectrum of compounds, but that's a separate video. Bacteria run a different metabolic chain entirely, and their end products are mostly organic acids. Lactobacillus, the bacteria behind sauerkraut, yogurt, and cheese, produces lactic acid. That's why these foods taste sour. Acetobacter produces acetic acid, which is why vinegar is vinegar. There are more exotic cases too. Some microorganisms produce hydrogen, xylitol, or compounds used in pharmaceuticals. But organic acids and alcohols cover the vast majority of fermented foods and drinks you encounter every day. Now here's the part that most fermentation content completely ignores, and it's actually the most important one for flavor, especially during yeast fermentation. Yeast don't just need energy. Like us, they need nitrogen and other nutrients to build new biomass, new cell material. To get these nutrients out of whatever you're fermenting, the yeast produce enzymes that break down the fruit or vegetable. In a very real sense, fermented food is pre-digested. The microbial enzymes do the work your gut would otherwise do. But here's what happens with the nitrogen specifically. Early in fermentation, when nitrogen is freely available, the yeast cells use it to grow and multiply rapidly. During this phase, they're producing very little in terms of interesting aroma compounds. They're just focused on growth. So what happens when nitrogen runs out naturally? The yeast cells start breaking down their own stored amino acids to recover the nitrogen inside them, a process called autophagy. When an amino acid gets broken down, the nitrogen is stripped out and the carbon skeleton that remains gets excreted. These carbon skeletons are higher alcohols. Isoamyl alcohol, for example, has an intense banana aroma. That's the banana note in a good wheat beer, not an additive, a direct byproduct of the yeast essentially digesting itself. These higher alcohols eventually accumulate to levels that become toxic to the yeast cell itself. So the yeast cell does something clever. It combines them with organic acids to form esters, less toxic, easier to manage internally. And for us, esters are some of the most important aroma compounds in fermented food and drink. Fruity, floral, complex notes. Most of them come from esters produced this way. And this has a direct practical implication for anyone fermenting at home. Adding large amounts of nitrogen to your fermentation, something some people do to speed things up, actually works against you aromatically. If the yeast always have enough nitrogen available, they never need to break down their own amino acids. And if they never break down their amino acids, you never get the higher alcohols and esters that create complexity. More nitrogen often means a cleaner but flatter result. Bacteria go through a similar nitrogen cycle, but the aroma story plays out differently. Lactic acid bacteria also break down amino acids under nitrogen limitation. But because their overall metabolism is far more streamlined, the higher alcohol and ester production is minimal compared to yeast. Bacteria's main aroma contributions come from other metabolic pathways. Diacetyl and acetoin, for example, are byproducts of pyruvate metabolism, the same central pathway that produces lactic acid. Depending on the bacterial strain and fermentation conditions, these compounds can give fermented foods buttery or creamy notes. Some bacteria also produce sulfur-containing compounds during amino acid breakdown, which are responsible for the sharp, pungent character of aged cheeses. The key difference from yeast is that in bacteria, these aroma compounds are less a result of nitrogen stress and more a natural byproduct of their core metabolic pathways running under specific conditions: temperature, pH, oxygen availability. We cover how these fermentation parameters influence the final product in another video. And the crucial point that ties all of this together: every species of microorganism produces a different profile of these compounds. Different higher alcohols, different esters, different ratios. That's why Lactobacillus plantarum makes a different sauerkraut than Lactobacillus brevis. That's why one wine yeast makes a buttery Chardonnay and another makes something crisp and mineral. And it goes even deeper than species level. Different strains within the same species produce meaningfully different concentrations of the same compounds. This is why professional brewers and winemakers obsess over strain selection, and why in a future video, we're going to go deep on non-Saccharomyces yeasts like Metschnikovia pulcherrima and Pichia cloyveri, and what they can do that standard brewing yeast simply can't. If you found this useful, the next video covers exactly that. And if you want to go deeper into yeast metabolism and fermentation science, I'm working on a detailed guide that covers everything we touched on today, and a lot more. Link in the description when it's ready.